Now we will discuss the last question, that is LAQ 21, tissue culture. It's also one of the most important LAQ. What is tissue culture? Here, growing or maintaining, or rather culturing, growing, it is growing, culturing and maintaining. Growing, culturing and maintaining of what? Maintenance of what is done over here? Maintenance of cells or tissues or organs in vitro. In vitro is called as tissue culture. Tissue culture is, or you can say that, growing, culturing and maintaining of cells or tissues or organs in vitro is called as tissue culture. What is in vitro? In vitro is nothing but inside a test tube or inside a petri plate, we can say, or under the aseptic conditions. Here, what is our important over here? The important terminology what we have to go with is that explant. Explant is the plant part, any part of the plant. Explant is any part of the plant which is taken out from the plant and it is grown under aseptic conditions on a nutrient medium. So, explant can be defined as any part of the plant which is taken from the, or which is isolated from the plant, and this is called as explant. This explant is taken, it is sterilized and it is grown into an full-fledged plant, or you can say it is grown into an individual or full-fledged, or you can say, or you can also call it as into an complete plant. So, here the explant under aseptic conditions, it is grown into an individual or a complete plant or a full-fledged plant. It is grown into a full-fledged plant in the nutrient medium. It's grown in the nutrient medium. Okay. So, this is what the entire thing. And this explant which is going to grow into a complete plant or into a full-fledged plant, it is called, that is purely based upon the technique that is called as totipotency. Totipotency is the capability of a cell to divide and multiply and to grow into an complete form is called as totipotency. So, these are the terminology which we must be aware of. And tissue culture is growing, maintaining, culturing of the cells or tissues or organs in vitro is called as tissue culture, wherein what we require is that explant is required over here. Explant which is taken out from the plant and it is grown on a nutrient medium under aseptic conditions, which grows into an full-fledged plant body or into an full-fledged, or rather into an complete plant. All this technique works under the principle that is totipotency, wherein a single cell is going to divide and redivide to form a number of cells or to form a complete plant. That's what. And different steps which are involved under tissue culture. The first step is preparation of culture medium. Preparation of the culture medium. The second step is sterilization of this medium. The second step is sterilization of the culture medium. The third step is inoculation. Inoculation of what? Inoculation of explant. And the next step is incubation. incubation of the explant. And the last step, that is acclimatization of the plantlet which is formed. Acclimatization of the plantlet which is formed. So these are the 5 steps under tissue culture which we have to discuss. First is the preparation of the culture medium. Next is sterilization of the prepared culture medium. The third step is inoculation of the explant. Third step is incubation of the explant, and the next 5th step, the last step, is acclimatization of the plantlets. Acclimatization of the plantlets. So what is going to happen into this? What are these steps? So first and foremost step which we are going to discuss here is preparation of culture medium. How the culture medium is prepared? Here a culture medium has to be prepared, and the culture medium must be a rich source of carbohydrates. Rich source of carbohydrates, the carbon sources, or you can say must be a rich source of carbon. Along with that of carbon, you can say the other things which has to be mixed within this culture medium, or which has to be present with this in culture medium, are, along with the carbon sources, this is hormones, amino acids, vitamins, all this. Okay, so the culture medium is composed of all these nutrients. Okay, the hormones, amino acids, vitamins, sugars. Okay, so now the culture medium is prepared in a distilled water in which all these are mixed, and now the culture medium is kept aside. As the culture medium is rich in nutrients, the culture medium, it attracts the growth of the microorganisms. As it attracts the growth of the microorganisms means wherein microorganisms can easily grow upon this medium which contains all these nutrients. So in order to kill the microorganisms which has already been invaded, or in order to prevent the invasion of the microorganisms to grow upon this nutrient medium, the culture medium has to be sterilized. This sterilization step is done. Sterilization of the culture medium. Sterilization of the culture medium is done in an autoclave. This is an microbiological laboratory equipment called as autoclave, wherein the culture medium is sterilized, or it is steam sterilized, you can say. Steam sterilized at 121 degrees centigrade of 15 minutes of time duration and at 15 pounds of pressure. So, at first we have prepared the culture medium, which consists of all the carbon sources, the vitamins, growth hormones, amino acids and sugars. As it is rich in nutrients, it has to be sterilized. Sterilization is carried out in an autoclave. Autoclave is an steam sterilizer in which the temperature is maintained of 121 degrees centigrade, which is sterilized for about 15 minutes of time duration at 15 pounds of pressure. So as the nutrient medium now will be sterilized in an autoclave. So at the end of the second step, we will be getting, or will be resulting, an nutrient culture medium which has been sterilized. That means which proves that there are no microbes. Okay, So that was happened. Now this sterilized nutrient medium is taken and explant is taken now. Meanwhile the explant is taken. Explant, as we have discussed earlier, that explant is any part of the plant which is taken. It is sterilized, external sterilization. External sterilization in the sense, the part, whichever the part, whether it is a part of the leaf or part of the stem, okay, any part of this is taken and it is surface sterilized, that is, it is washed in detergents. So as it is washed in detergent, again this is also done in order to ensure that the explant is also microbial free. Now this microbial free explant is inoculated upon the sterilized nutrient medium. This inoculation is done, this inoculation, where it is done, this inoculation, it is done under laminar airflow chamber, under laminar airflow chamber. This gives out aseptic conditions. Aseptic conditions. Aseptic conditions. Aseptic conditions are again microbial free conditions. Okay. So the explant which has been sterilized, external sterilization or the superficial sterilization by means of the detergents, now it is inoculated onto the sterilized nutrient medium under aseptic conditions, that is, which is done in an laminar airflow. Laminar airflow is one more laboratory equipment wherein which has an UV light, under UV light which is of totally microbial free. Now after inoculating the explant into the sterilized nutrient medium, the explant is kept on the nutrient medium as such for about 3 to 4 weeks of duration. That is called as incubation of explant. Incubation is the period, duration period we can say. Incubation is the duration period wherein after the inoculation of the explant, the nutrient medium having the explant is kept aside. The nutrient medium having the explant is kept aside for about, kept aside in the sense, kept in the laboratory itself for about 3 to 4 weeks. So it is kept for about 3 to 4 weeks. That is called as incubation period. So when it is kept for 3 to 4 weeks, what happens? So here, then after the incubation time, what happens? Slowly you will see the appearance or the proliferation of the tissue. Proliferation of the tissue. Proliferation is nothing but protruding out from the nutrient medium. After the period of incubation, the explant, whereas under the technique or under the principle that is called as totipotency, wherein the explant divides and forms multiple cells, which results in the formation of proliferating mass of undifferentiated cells. This proliferating mass or proliferated mass of undifferentiated cells is called as callus. Proliferating mass of undifferentiated cells is called as callus, which is seen upon the nutrient medium. So because the explant is going to absorb the nutrients which are available in the sterilized nutrient medium, and under the principle of the totipotency, the explant is going to divide within this incubation period of 3 to 4 weeks, wherein it is going to form this proliferating mass of cells or tissue called as callus. Okay, now if at all this callus is shifted on to the hormones or the medium containing hormones, now from the callus, if at all plant growth hormones, plant growth hormones, if we go with, that is auxins or cytokinins are added, Now what happens? Then we can observe the formation of or initiation of root and shoot. Formation or initiation of root and shoot is observed. So due to the addition of or by the addition of plant growth regulators or hormones like auxins or cytokinins, which lead to the initiation of the formation of root and shoot. This initiation or the formation of root and shoot is called as organogenesis. What is it called as? It is called as organogenesis. That is the formation of organs. Okay, so this what can happen in one case. For sometimes the other case can also happen. What is that? Is that the callus may also result in the formation of, or which results in the formation of embryo. Okay, so callus results in the formation of, or from the callus embryos develop. This process of development of embryos from callus is called as embryogenesis, and these embryos which are formed from that of the callus are called as embryoids. They are called as embryoids. Okay, these are also called as somatic embryoids, and the process is called as somatic embryogenesis. Got it? So the callus, when it is kept on a plant growth regulators which helps in the initiation of the root or shoot, this process is called as organogenesis. In some other cases, from the callus embryos also develops. This is called as embryogenesis. These embryos are called as embryoids, and these are called as somatic embryoids because they are going to develop from the vegetative part of the cells. That's why these are called as somatic embryoids. So that's what over here under case of the callus where it is going to develop. Now embryos develop, okay, whatever it is. Now what is going to happen? Now formation of organs. These organs further they are going to give rise. Now these organs or the organs by means of organogenesis, root and shoot formation takes place, we have gone with. So it is further going to develop into a full plant. Now the last step comes over here, that is acclimatization of plantlets. So now the plantlet which has been developed upon the nutrient medium, the plantlet which develops on the nutrient medium, slowly the nutrient medium is washed out with distilled water, and this plantlet is transferred to an material or transferred to that of the material, basic material containing soilrite. What does it contain? It contains soilrite. Soilrite is a material or it is a composition consisting of coconut shells, plus other composed of organic material, So now the plantlets which has been developed as a result of organogenesis, wherein they are transferred now into the soilrite. Soilrite is a composition of the coconut shells and organic material. Now, and a poly bag is covered, or poly paper. The plantlet is covered with a poly paper, so that this wrapping in this poly paper provides humidity. It provides humidity. So due to these plantlets are placed as such, placing in the soilrite covered with that of the poly paper, they are placed at room temperature, within the labs itself, at room temperature within the labs. Okay, so then later after some time or after some period, it will be observed that the plantlets have gained somewhat firm, looks to be somewhat firm, so that then these plants will be transferred finally to the fields. Will be transferred finally to the fields. That is comes under the acclimatization of the plantlets. Acclimatization is nothing but adjusting of the plantlets to that of the natural environment. Now if we go with that of the flow chart over here of tissue culture, so flow chart of the tissue culture, if we go with, it is nutrient medium, that is preparation of nutrient medium, or you can also call it as culture medium, plant. Okay, the culture medium is now, it is sterilization of this culture medium. Now from the plant, what is taken? Explant is taken. Okay, now what happens now? Okay, now the sterilized, within the sterilized nutrient medium, the explant is inoculated. So inoculation of explant, inoculation of the explant on the sterilized medium. Okay, that is which is done under laminar airflow chamber. Sterilization is autoclaving. Okay, and the temperature here is 121 degrees, 15 minutes of time duration, 15 pounds of pressure, 15 pounds of pressure. Okay, now after inoculation of the explant, then incubation period, and the incubation period is 3 to 4 weeks. Incubation period is 3 to 4 weeks. After incubation, what happens now? Okay, incubation, organogenesis, that is formation of root or shoot, formation of the root or shoot, organogenesis. Then transfer into the medium so that plantlet is formed. So I am concising this flow chart. This is actually a flow chart which shows tissue culture. A flow chart showing the tissue culture. Okay, plantlet, where the plantlet is formed. Sometimes after the organogenesis, what happens here? What is formed? Which results in the formation of embryoids. Embryoids are formed, which is nothing but called as embryogenesis. Okay, then so now the plantlets are now transferred to pots. That is what you can say, it is acclimatization into pots and then into fields. Acclimatization into pots, then into fields. Okay, so that's what. Okay, the embryogenesis over here. Okay, plantlet is formed, and here organogenesis wherein the root and shoot plantlets are formed, which are transferred or to the acclimatization, that is into the pots, then into the fields, embryos, embryogenesis. And these embryoids can be, sometimes they are encapsulated with sodium alginate solution. Sodium alginate solution. Now, the embryoids which are encapsulated with, or the plantlets which are encapsulated with sodium alginate solution, are called as artificial seeds. Artificial, or rather they are called as synthetic seeds. Okay. So, during the matter of the transportation, it can be utilized. So, that is what, wherein the preparation of the culture medium, the flow chart is here with preparation of the culture medium, wherein the culture medium is sterilized in an autoclave. Meanwhile, a plant, an explant is taken, and this explant is also sterilized. Okay. And inoculation of the explant upon the sterilized medium, incubation. Then actually, to say here, we can go with that of callus formation. From the callus, organogenesis, formation of the root or shoot, which develops into the plantlet, then develops into the, and which are transferred to that of the pots. And sometimes from the callus, you can say from the callus, development of the embryoids, which are called as the somatic embryoids. Okay. These plantlets which can be encapsulated by sodium alginate solution, which are called as artificial or synthetic seeds. For transportation, it is good. So, that is what this is, the flow chart which shows the entire tissue culture technique. Through tissue culture, what are the beneficial aspects, or what is the beneficial aspects of this tissue culture? You can say that micro propagation is one thing we can go with. Micro propagation. Wherein within the limited time and limited space, okay, within short duration and within short time or within short space, more number of plants can be developed. That is, micro means small, limited, propagation means to develop. So, they can be developed, that is called as micro propagation. And the plants which are produced are somatic plants. That means which develop from that of the vegetative cells, purely from that of vegetative parts of the cells. That's the word soma clones we say. Soma word refers to vegetative cells. Clone word refers to the which are exactly identical to that of the source plant from which it has taken. That is soma clones. So, micro propagation, one term, yes, it is useful in, and it is also useful in producing soma clones. And meristem culture is very important. Meristem culture is why? Because if meristem is taken which is totally microbe free. So, that is what comes to the end of tissue culture.